And with that, I'd like to introduce Dr. Jamie McFarlin. It's my real pleasure to have her here speaking with us virtually today as part of our interviews for this faculty position in Earth and Planetary Evolution. Jamie got her bachelor's degree at Lawrence University in Wisconsin in geology and environmental science, and then went on to a master's and a PhD at Northwestern, that's a little bit to the south, Chicago area, Earth and Planetary Science Department there. And she won herself a NSF graduate research fellowship that supported some of her time there. She's now a postdoc at CU Boulder, working with Sebastian Kopp there on some neat molecular geochemistry work. Her work has largely focused on using geochemistry, organic geochemistry of various different flavors to reconstruct, Holocene and pre-Holocene climate conditions and environmental conditions and biogeochemical processes in cool regions of the Earth. Specifically, she's done a lot of interesting work on lake sediments in Greenland, which I think we'll hear about some today. I noted that she originally went into her PhD program ready to work on insect fossils and did some work on that. Got a PNAS paper out of that before she moved on and was swayed to the dark side of organic biogeochemistry, which is a big focus, I guess, or one of her primary tools now. She's really contributed substantially to the organic geochemical record of Greenland, Greenland's climate history, both in terms of developing methods that are innovative, as well as developing climate reconstructions, and then linking those to biogeochemical processes. So looking at the whole kind of climate change, biogeochemistry, climate change feedback loop to some degree. In her postdoc, she's working on isotope ratios, measuring isotope ratios of GDGTs, which are these really hot, maybe a bad pun, really exciting compounds that are being widely used now in paleoclimate reconstruction, but largely just looking at their abundance. And she's trying to work to add isotope in the mix there. And she also has interest in the techniques of stable isotope probing, which kind of the emerging set of tools to understand ecosystem processes or biogeochemical processes, looking at stable isotope signatures in the modern. The last thing I'll say about her is, in addition to all this great science, kind of innovative work she's been doing, she has, or for her career stage, I think a really strong record of teaching and mentoring. So at Northwestern, she in particular had one undergraduate student that she basically took from start to finish, well, not finish, I guess, launching the next stages. A student who, during the course of their undergraduate work with Jamie, published a paper and is now off as a grad student at Caltech. And then she's contributed to teaching, I guess, at both institutions. She's been at PhD institution and now at Colorado, where she's actually instructor of record, co-teaching a class on global change there this semester. So I've taken up enough time. With that, I'll turn it over to Jamie. If you want to throw up your slides, take it away.
Yeah, thank you for that introduction, Gabe. That was a really flattering overview of a lot of the things I've worked on. So I'm just going to go ahead and share this really quickly and then move it into view. So I just want to make sure that you guys can see my slide view and not the speaker view. Is that correct? Okay.
Looks good.
Thank you for that feedback. Okay, well, thank you so much. I'm absolutely delighted to be here to give a talk. And in this talk, I'm going to kind of walk through three major themes of research. So I'm going to kind of talk about what we can learn from geologic records about the relatively near-term impact of warming on Arctic landscapes, things we might expect to see broadly develop during the middle to end of the century, given ongoing modern warming. I'm also going to talk about what's new in the world of hydrogen isotopes. So I'll demo some major updates to proxies that are widely used to reconstruct past water isotopes. And then finally, I'll talk about what's at the forefront of compound-specific isotope research, so where I think the field is going and some major areas for growth. And these are points that I kind of talked a bit about in each of the meetings today, so they might be familiar to a few of you. So where I kind of want to start, though, is just with this plot of ongoing modern warming because I think it speaks to how much the planetary energy balance is changing and how rapidly. So here I'm plotting average global temperatures over the last 50 years. And these are observations of average surface temperatures that show that globally, the average surface temperature has risen about a single degree Celsius. If I restrict the data, though, to just the northernmost latitudes, so from 60 to 90 degrees north, over the last 20 years, especially, we see a 2 to 3 magnitude increase of change relative to the global average. So 2 to 3 times the amount of warming is occurring in the Arctic relative to the global average. So that means that the Arctic is sort of a huge weight in this global trend. That's really important to think about because essentially how goes the Arctic, so goes the world. Feedbacks in the Arctic greatly amplify regional and global warming. So here I'm highlighting feedbacks that derive from declining sea ice. And sea ice is really one of the first major things to change as the Arctic warms. A reduction in sea ice not only impacts the global energy budget through declining albedo, so a decrease in the reflectivity of the planetary surface, but it also impacts the global latitudinal temperature gradient because sea ice retreat increases the flux of ocean heat to air, particularly in the fringe seasons. And just a sort of interesting and maybe scary projection is that the best estimate we have for when we're going to have no more annual sea ice is between 2045 and 2060. There are also major stores of organic carbon in permafrost in Arctic lakes that are currently frozen or otherwise preserved that are vulnerable to respiration once thawed, meaning that they get consumed, metabolized, and then emitted as CO2 or methane. And that's potentially a huge flux of greenhouse gases to the atmosphere. And then there's also potential for increased freshwater flux into the North Atlantic due to increased precipitation over that area of the Arctic, which is broadly projected by climate models, and also due to the melting of land ice, which has the potential to weaken the thermohaline current, at least temporarily, which would have major changes on global circulation and the distribution of energy. So these are all things that we're starting to see at least whiffs of in the Arctic already, although there's huge uncertainty in how much and how fast things like frozen carbon reservoirs or precipitation patterns are going to change over the coming century. We know very little about the sensitivity of these landscapes to rapid warming. So there's this urgency to understand as much about this landscape as we can, given the rate of modern warming. So how do we get at understanding how these landscapes change as they warm? I think a Venn diagram is kind of the best way to summarize this. So I'm going to show you the 3 major approaches and how the scientific community really works together at understanding global change in sort of equally important ways. So one approach is through modern observations. The next is through model simulations. So trying to force models with warming or cooling and seeing how the system responds. And then the third approach is to use geologic records that really capture past climate periods. And my emphasis is on Arctic warming. So all 3 approaches are really necessary for increasing our confidence in approaching a scientific consensus and what we predict a warmer world will look like. And within this Venn diagram, my research focus is really on using geologic records and modern observations. And one advantage I would say that's distinct to geologic records and modern observations is that they can show us information that we really can't capture in models, things like abrupt change and biologic feedbacks. So my overarching motivation as a scientist is to understand how Arctic landscapes change as they warm and how those changes propagate into global systems. This is clearly a very broad research focus, and that's because I tackle a variety of aspects of environmental change using a wide array of tools. I pull tools primarily from isotope and organic geochemistry, but increasingly from geobiology. For this talk, I'll focus entirely on one aspect of my research, which is an effort to use hydrogen isotopes and biomarkers to reconstruct hydroclimate dynamics across Greenland during the Holocene. Although where that story ends is somewhat surprising. So the Holocene is really our best opportunity to look at a past period of Arctic warmth from terrestrial records in really high spatial and temporal resolution. Typically, Arctic terrestrial records don't extend much older than the Holocene due to widespread glacial erosion during the last glacial maximum. And although the Holocene isn't what I would describe as a period of extraordinary global change, orbital cycles drove warmer summer temperatures in the early and middle Holocene across the Arctic. So here I'm going to give you a frame of a plot where I'm going to demonstrate how much warmer things were over Greenland during the Holocene. So in this plot, I have time on the x-axis going from oldest to youngest in thousands of years before present. And every plot I'll show you today is going to be on that axis. On the y-axis, I have temperature change in degrees Celsius. That 0 line is the pre-industrial average, and that dotted line is showing the modern Arctic average temperature anomaly. So the first plot I showed you showing Arctic temperature change, the endpoint of that plot would hit that dotted dash line. Okay, so now to this plot, I've added a record of temperature, and this record derives from oxygen isotopes in the Redland Icecap, which is located on East Greenland. And it's thought to be a record of mean annual air temperature that's amount weighted. This record starts at the end of the Younger Dryas. And actually what I should say is I've plotted the start of this record at the end of the Younger Dryas, although the actual isotope record goes back through the last glacial period as well. But I'm plotting just the Holocene here. And so it starts at the end of the Younger Dryas, which is sort of the last whiff of cooling before we move into full Holocene warmth. And that marks the start of the Holocene period. What you can see is that from 10,000 to 5,000 years ago, temperatures were about as warm as our modern anomaly, but that period of warming lasted for thousands of years. If you compare that to modern data, we have observations of warming for about 10 or 20 years. So this gives us a much longer time series to start looking at how things change when you expose them to sustained continuous warming. So this is just the average inferred from an ice core. But what happens when we look at more seasonal summer-based proxies across Greenland? So in blue, I've now added a record from West Greenland, and this record was produced using a Chronometer species assemblage temperature function. And so this is taking a two-wing fly that has a larval stage, pulling the larval head capsules out of sediments, using taxonomy to look at the community composition, and then using a transfer function that relates community composition to temperature. And we're going to come back to chronometers later, but that's essentially where these temperature records are derived. It's a thermophilic species, so species that's temperature dependent, and their distribution is related to temperature in an empirical way. And the error on that model is over here. So that's our chronometer inferred error. So what you can see in this lake, so you can see on the map, I've added it as a little blue point in the west part of Greenland, is that it agrees pretty largely with the isotope record for at least as far back as this record extends. When I add another lake from East Greenland, we see a greater amplitude of warming in that lake in the early part of the Holocene. So from about 10 to 6,000 years ago, warming in the early Holocene during the summers is actually looking like it was reaching maybe 4 to 5 degrees above modern temperatures. And if we go even further north, using this exact same proxy, so this is a lake called Waxlips Lake, it's in northwest Greenland, we see an even stronger amplitude of warming at this site. So now we're looking at upwards of 5 degrees of warming in the summer in the highest continental latitudes of Arctic regions. So this is actually a really huge amount of warming for something in a global sense didn't really change as strongly as other periods in the geologic record. So the Holocene really presents this incredible opportunity to examine the impact of large changes in Arctic temperatures that roll out over tens to hundreds to thousands of years. So one major question in this is, how did warming impact precipitation patterns across Greenland? Although climate models indicate broad increases in precipitation should occur with warming across broad swaths of the Arctic, the geologic record of past precipitation is a lot more complex. In blue, I've overlaid one of the longest, highest resolution records that we have. This is of precipitation on Greenland, and this record is of accumulation at the Gist 2 ice core site in central Greenland. The most notable feature of this record is the doubling of accumulation that occurs at the onset of the Holocene. This doubling is thought to have occurred in as little as a couple of years. So from this record, it's clear that the hydrologic system responds immediately to temperature. But this is just what's happening over the ice sheet. So what do we see in ice marginal records where changes in precipitation really matter to ice sheet mass balance, because that's where ablation zones are. So now I plotted the few records we have on Greenland from the Holocene that infer some aspect of hydroclimate, and most of these simply get at precipitation evaporation balance of lakes. So as purple stars, I plotted lakes where the proxy indicates a wetter Greenland during the early Holocene than the late Holocene, and as yellow stars, I've plotted lakes where proxy information indicates a drier early Holocene than the late Holocene. You might notice that the neighboring records in West Greenland, but really in all of Southwest and East Greenland, these neighboring records show contradictory trends. And it's pretty unclear right now if that's because there's very strong spatial heterogeneity, or if it's because the proxies that were used in some of these records aren't actually doing a very good job of capturing precipitation and evaporation balance. So it seems like a simple question, but it's an important one to ask. Why do we care about how hydroclimate has changed in the past and how it might change in the future? Well, we care because water is key to understanding the impacts of climate change on a multitude of systems. These systems include landscape dynamics, ecosystem dynamics, humans, society, and health, and of course, climate feedbacks that are embedded in the hydrologic system. So I'm again using a Venn diagram. I really like them for summarizing information, and I'm going to use it to show the ways in which water really matters. So I've added just a few examples of aspects of Arctic landscapes that are sensitive to changes in the hydrologic cycle. As an example, if more precipitation falls as rain in an Arctic region, it's likely that permafrost is going to thaw faster. And it's also likely that land ice is going to melt faster, which will change planetary albedo and impact climate. Another example is that water availability really modulates vegetation structure and can impact soil erosion. And so those are just more examples of the way in which water kind of sits at the nexus of all of these systems. Another example of how a specific and sort of understated change might have cascading consequences is if you increase precipitation, you might have more standing water in the summer. Standing water has a tendency to have anaerobic metabolisms, especially in the sediments, and anaerobic metabolisms often respire methane instead of CO2. And so you might increase methane emissions from these water bodies. The increase of methane emissions is going to have a climate feedback because it's a potent greenhouse gas. And so you might have temperature warming and more standing water, both working to expand the range of mosquitoes from where they currently persist into more northern latitudes. And that might spread the range in which mosquito-borne diseases are able to impact human populations. So that's just one sort of connected system in which we can think about water as the center of it, but there are, we can think of probably an infinite number of examples. So water is important, but despite how important it is to observe changes in the water cycle over continents, it can be a really challenging aspect of the climate system to reconstruct, especially in the Arctic. We can look to a number of existing tools to try to reconstruct water, but of course, all of them have pros and cons. Ice cores are an absolutely incredible archive of precipitation, but they're spatially limited. They show us conditions over the ice sheet, and these may be fundamentally different to conditions that are even just marginal to the ice sheet. Macrofossils and sediments often relate to indirect aspects of water balance like salinity, but the signal they record can be complicated by competing environmental gradients and sometimes lag true changes due to migration, history, and colonization time. Sedimentary structures like the mud cracks I have pictured here are inconsistently preserved and not available from Holocene age to lake sediments. So what we increasingly look to reconstruct precipitation across the Arctic through geologic periods are isotope proxies. So biomarkers whose oxygen or hydrogen isotopic composition are empirically related to that of their growth water. And that's based on modern observations. Isotopes are a really powerful tool because they are process-oriented. And in water, isotopes really reflect hydroclimate processes. So most simply, they reflect primarily temperature, evaporation, condensation, and transport history. These processes are so consistent in how they distribute isotopes, we can predict the average isotopic composition of precipitation pretty well for really broad spatial regions. And here I plotted the line that describes the relationship of hydrogen and oxygen isotopes in precipitation globally. So this is Harmon Craig's global meteoric water line. That's abbreviated on this figure as GMWL. Regions that are highly evaporative might fall off this line, which I've kind of shown with that little orange annotation. And that's because the rate differences during phase changes between oxygen and hydrogen can change the slope and the position of it owing to the proportional mass size differences between those elements and their isotopes. And I'm going to be talking about isotope ratios throughout the rest of the talk, and I'm going to be talking about them as delta notations. And so I kind of put a little isotope 101 up here for those of you that are unfamiliar with isotope, isotope delta values, and just the lingo around isotopes. But really what you need to know is that moving along either axis relates back to hydroclimate processes. So on this plot where I plotted hydrogen and oxygen delta values, what you can think of is as you increase, so you enrich those isotope values, and on the y-axis that would be moving up, and on the x-axis that would be moving right. That relates probably either to a warmer season or a warmer region. It can sometimes relate to evaporation if you only have one of those two isotopes that you're looking at. If you're moving the other direction, it's going to relate probably to a colder season or a colder region. Or you could additionally have input from a different reservoir of water, like groundwater, that can change where you're sitting in this water space. So I've now added to the side this fantastic figure from waterisotopes.org. So this is work housed there at the University of Utah, led by Gabe Bowen. And this figure shows modeled annual average hydrogen isotopes and precipitation across North America. And you can imagine if you were to pick a point anywhere on this map and reconstruct precipitation isotopes through time, if those precipitation isotopes show major changes through time, it could be related to broadly changing isoscape, which speaks to changes in large-scale circulation and climate patterns. Or it could just be a regional signal. So we really need to build sort of an extensive past data set on water isotope records to tease out a regional signal from a broad-scale change. So across the Arctic, lakes are really the key to building spatially widespread and high-resolution records of Holocene warming. Lake cores are absolutely beautiful archives that act like a library that holds information about environmental conditions that are cataloged by time. We can radiocarbon date organic material, create a chronology, and then extract biotic or abiotic information depending on the study goals. So I've kind of shown you an abundance of some of the common types of things we can pull out of cores, like macrofossils, elemental abundances, and organic compounds. And of these things, when I work on lake records, I get very excited about lipid molecular fossils. So these are essentially fat compounds that are non-soluble in water. Just for fun, the ones I plotted are branched GDGTs, an alkane from a plant wax, and then a crenarchaeol from an archaea. There are literally tens to hundreds of compounds that accumulate in sediments, and I really only have time today to talk about hydrogen and the hydrogen isotopic composition of some lipid compounds. The lipids offer structural information and carbon isotope information that relate to the origin of organisms, the productivity of the system, and all sorts of other environmental factors. And I study all of those aspects. So I'm going to focus in on just one class of lipid compound and talk about sedimentary plant waxes. So these are really simple hydrocarbons that are globally ubiquitous, easily extracted from sediments, and the hydrogen isotopes of plant waxes primarily record their growth water. The traditional framework by which we understand and use this proxy is that we measure the plant wax hydrogen isotopes, so that yellow box, and then we use those to track water because we expect that they're offset by their environmental water by a predictable amount. And that predictable amount is called the apparent fractionation. They're also source specific to a degree, and so we can infer whether or not that wax compound came from a terrestrial plant or an aquatic or a non-vascular plant based on the carbon chain length. So that's the number of carbons in the carbon chain of that compound. And so terrestrial plants are biased towards long chain waxes, and I've abbreviated that as C29, with 29 indicating the number of carbon. And aquatic and non-vascular plants are biased towards shorter chain compounds in the mid-chain length, so a 23 would be an example. And then what we think that might tell us is that because they have different growth forms and grow in different environments, we might start looking at trends that are different between the growth waters. So in a terrestrial plant, you would see precipitation via soil water, and an aquatic plant would be growing with lake water. And so reconstructing differences in those changes might tell you something about the hydroclimate balance on a landscape. However, the fidelity of plant waxes in reconstructing precipitation in Arctic regions was largely unknown when I started working in the Arctic. So when I first started working on this proxy, I had a big question, which was, do plant waxes in the Arctic fit this conventional wax to water framework? And that question was motivated by the idea that plants in the Arctic have a very short growing season during which they're photosynthesizing 24 hours a day. They have to grow in an Arctic climate, which can be cold, it can be snowing on them, it can be very windy. There isn't always a lot of other vegetation around. And there was really limited data at that time to prove that this relationship could be extended to Arctic regions. And so as a PhD student, I developed a transect that included 22 lakes on the western coast of Greenland to really test this question. And so I extracted plant waxes from surface sediments because I really wanted to target this as a sedimentary proxy for reconstructing precipitation isotopes. And so I looked at surface sediments as the proxy calibration. And I pulled waxes from glacial, non-glacial, and evaporative lakes. And the reason I did that is because in glacial and evaporative lakes, the lake water isotope composition is going to decouple from precipitation. And so you can start to see if things that live in lakes are actually tracking lake water and not just precipitation, so that you don't have confounding signals. And this transect covered 16 degrees of latitude, which is a huge climate rate gradient. And I'm going to show you just some pictures of that to really give you a sense for this climate gradient. So this is a lake in Northwest Greenland. This is Waxlips Lake. This is me sitting on our coring rig with another PhD student at the time. And this is the Greenland ice sheet 2 kilometers behind us. And you can see there's very little terrestrial vegetation surrounding this lake. But what you can't see is that there's a very dense carpet of aquatic mosses blanketing the sediment surface. And it actually made it a real challenge to core this lake. And I'll show you down core data from this lake in just a little bit. This is a lake called Stardam. It's right on the edge of a fjord where you have icebergs calving. This is all in Northwest Greenland at 76 degrees latitude. This is our field camp. And now I've moved us down to West Greenland. So this is the Kangerlussuaq region, and you can see it is much different environmentally. So there are shrubs, it's very dry. This is an evaporative lake called Little Sugarloaf. And this region is very dry and very windy. And then finally, here's some shots from South Greenland. And what you can see here is that it's glacially carved. It's lush compared to the other parts of Greenland anyway. And these field sites, just to give you a sense of what field work looks like when you're working in Greenland, we get helicoptered in, our gear gets helicoptered in. So this is a helicopter that's pulling a load of gear. So this would be our coring supplies in our field camp. Here's me actually riding in that helicopter out to camp. And here's a picture of one of the camps we set up at that site. And what you can see in this picture is that it's very foggy, indicating it's a much more humid, moist climate where there's a lot more rain year round, especially in the summer. Okay, so once I had sampled 22 lakes, I came back to the lab and I actually extracted 11 different lipid compounds and measured hydrogen and carbon on all of them. And by hydrogen and carbon, I mean the isotopic composition of those elements. And I'm going to show you just the hydrogen data today. So here I plotted the hydrogen isotopic composition of the C29, so the terrestrial plant wax, and I plotted it against the hydrogen isotopic composition of mean annual precipitation as inferred by the online isotopes of precipitation calculator. And we use that as just an average, an average precipitation value for that region. And what you can see in this plot is that these regions And their plant waxes have a pretty strong relationship with the environmental water that's available on average. And so they plot kind of exactly where we would expect, with the most depleted values from the northernmost sites and the most enriched values from the southernmost sites. And so from that, we thought, okay, that's a pretty promising data set, but how does it look in a global framework? Like, if we start to measure hydrogen isotopes on these plant waxes, can we compare them to a site in Wisconsin, or can we compare them to a site in Peru and still have it be meaningful? So what I did is I went to the literature and I compiled every data point I could find from sedimentary systems. And so these are only lake systems where people have pulled plant waxes and measured the hydrogen isotopes on them. And so I compiled it into this global framework, and this now includes about 200 sites from latitudinal range of 0 to 80 degrees north. And once I put the Greenland data into this compilation, what I saw is that it actually fits and nests really tightly into this beautiful global framework. Another way you can look at this data is to do this as an average difference in per mil space between the plant wax and the growth water. And so that's called the epsilon, or the apparent fractionation. And the value that we got when we added all this data together was 121 per mil. So plant waxes are on average 121 per mil more depleted than their growth water. What's really fascinating about that number is that is the original number that the first study that did this looked at, but with just a fraction of the data points. So we added over 100 more data points into this compilation, and the apparent fractionation factor didn't change at all. So that tells us that there's actually this immensely robust and predictable relationship between these plant waxes and precipitation globally. This is a growing and always developing project. So as more data becomes available, we will add more data to it. So if you are a person who works on this or know somebody that is and has new data, I would love it to be submitted to my OSF database. And this is just intended to serve as a community resource, and it actually has all 11 of those compounds I measured in there. So today I'm just kind of showing you the end members of those compounds. We did that same test, though, for the aquatic plant waxes, so the C23 alkanes, against lake water specifically. And remember, we had a transect that had lakes that decoupled from precipitation. So if it was truly growing using aquatic water, we would expect that it would track lake water instead of precipitation. And what we saw when we did that is that actually the relationship between aquatic plant waxes and lake water is fairly weak. You can see our lake water only spans approximately 40 per mil for a lot of our data for the bulk of these sample points. And yet our plant waxes cover a range of more like 100 per mil. And it's non-discriminatory by the type of lake. So this is all these different types of lakes. And yet the plant waxes do not really correlate strongly to that lake water. So that would be the point where we felt like, okay, we kind of broke this proxy, but we will build it back up in a different way. And I'll show you that in just a minute. As a sort of random aside, I'm going to really quickly jump to just a note that I think there is a lot of potential to do more to develop water isotope proxies in around Utah, because there is this beautiful aridity gradient around the Southwest. And so here I've just plotted two sort of more simple targets, and I could imagine expanding this into other more complicated biomarkers. But I have sort of this idea that you can do paired oxygen, hydrogen to start getting at more complicated signals within water isotopes. And the best place to really do that is looking at it on a aridity gradient where you have a strong signal. And this would be a, sort of a project that I think would be really fun to involve local students in field work and in isotope geochemistry and in lake coring and lake and limnology. And it's just has the potential to start small and grow big. So I'm going to jump back to Greenland now. Okay, so I felt like that I was pretty confident that plant waxes at least recorded precipitation across Greenland and could be used to reconstruct precipitation through the Holocene. So while I was collecting my transect samples, I also cored multiple lakes. And so I'm going to show you some downcore Holocene data from those lakes now. And so Greenland is unique because we have these ice cores that give us a baseline for how precipitation changes. So the first thing I've done is just plot an ice core where we've actually got ancient precipitation that can be sampled for water isotopes. And so this is the ice core oxygen isotope trend line that is from Redland in East Greenland. And it's plotted from 10,000 to 0 on the thousands of year before scale. And I'm going to add 3 lakes to this. So I'm going to add a lake in northeast Greenland called Trimpesow. I'm going to add Waxlips Lake, which is in the northwest of Greenland. I'm going to add Lake N3, which is in West Greenland. And this N3 is the only data set that I did not personally measure. It is from published data that is available in the literature that use the same proxies. You can see each lake on my map to the left. And then just to orient you really quickly with the framework here, all of these wax isotopes, the calibration data was used to convert these into water space instead of wax space. And so what I've also added to this figure is these shaded regions are the range of modern precipitation isotopes in each of those regions today. And modern lake water that was measured during the field season from water that was collected during the field season. And just to further clarify, the top of this bar is going to show you summer water isotopes, so the heaviest isotopes you see in these regions. And the bottom is going to show you winter water isotopes, so the lightest ones we see in these regions. And as you move up the scale, you're probably looking at either temperature or evaporation. Okay, so in pink, I have now added terrestrial plant water, which is correlated as precipitation in these calibration data sets. What you can see in these, I'm going to add a bunch of squiggles now into the ice core plot, is that they broadly track installation-driven cooling, this multi-millennial installation-driven cooling that goes from peak warmth in the early Holocene towards cooler in the late Holocene. And if we put that into oxygen isotope space and overlay it on our Greenland ice cores, what do we see? Well, it's kind of similar to what we see in our temperature records, where we see the most amplified amount of change in the northernmost region. So we see the largest shift in our isotope signals at Trimpesow and the smallest at N3. And that adheres to this latitudinal gradient. Now, there is a summer bias nature of this proxy, potentially, although the calibration data suggests that this is mean annual precipitation. But where these isotopes plot in our modern water space is very strongly summer biased. And so this suggests if precipitation increased, it increased during the warm season, when most precipitation is delivered as rain, and that has implications for permafrost thaw. And notably, all sites are on the margin of the ice sheet where most ablation occurs during the summer. So this might help explain this rapid retreat at the margins of the Greenland ice sheet during the early Holocene. When I looked at the aquatic plant waxes, so the C23, what I found are these just absolutely massive excursions at all 3 sites. So there's 150 per mil change at Trimpesow between long chain waxes and mid chain waxes, 160 at Waxlips, and like 180 at N3. And that's an absolutely massive range. And it actually extends beyond the range of precipitation isotopes in this region by 50 to 60 per mil in each region, except Waxlips, where it is just beyond the range of winter precipitation isotopes. So I already was skeptical of what this proxy was showing us because of that poor correlation in our modern transect. But the fun thing about isotopes is that there's always another proxy and always another isotope. And so I also compared these data to oxygen isotopes from Chronometer. So let me just jump out of plant wax world and into Chronomets and kind of introduce you really quickly to one more proxy. So this is a chironomid. It is an insect, 2-winged fly, and they're globally ubiquitous and highly abundant. And in Arctic lakes, there are hundreds of the head capsules of their larvae. So here I've plotted a picture of the Chronometer larvae, and I've thrown a bone to anybody out there who likes to fly fish, because fly fishers tend to know what these are, even if they don't know what they are. When you're fly fishing with the beadhead, you're fly fishing with a Chronometer larva. Chronometer larvae go through 4 phases where they shed their exoskeleton, including these chitinous head capsules, and those accumulate abundantly in sediments. So here's just a few pictures of what those head capsules look like. These are head capsules I handpicked from sediment at Waxlips Lake that went into my temperature calibration at Waxlips Lake that was published in PNAS. For the oxygen isotope work, we don't do species-specific work. We just do anything we can find goes into the EA. But the important thing is that oxygen isotopes from chronometer capsules have a very strong relationship to lake water in the modern. So they're highly correlated to the oxygen isotopic composition of the lake water in which they grow, and they're obligate aquatic, so they're growing year-round in lakes. And in this calibration data set, there are lakes from Greenland, so we are pretty confident that this proxy works there just fine. And so this is actually a more confident, more robust proxy for lake water, given the calibration data we had from aquatic plant waxes. And so we used this at Wax Lips Lake, and it was fortuitous that it was also published for Lake N3. And so I'm going to now add to this figure of water isotopes chironomid oxygen that was transformed to water isotope values using that calibration data, and then further transformed into hydrogen using the local meteoric water line. So I'm going to add that as blue. And here's what those trends look like. So what we see in the chironomid larvae, which are these blue lines, is that they plot pretty much on top of our modern lake water. They track insulation-driven cooling. You can't really see it because the scale on this is so tight because of those huge changes in those mid-chain waxes. But they're relatively stable. And so now we have two proxies that are totally independent, the long chain waxes and the chironomid, that show that lake water and precipitation is tracking insulation-driven cooling. It's summer biased, and it is relatively stable compared to our aquatic plant waxes. And so I just want to really emphasize, like, the trends we're seeing in these aquatic plant waxes and hydrogen are absolutely massive. And we have an independent proxy for lake water that shows this lake water is not changing in that way. So how in the world do you take an aquatic plant, fill it with lake water, and have hydrogen isotopes decouple from lake water? So that was a huge question and a huge mystery that I was really excited to solve. And so what I did is I looked to other paleoenvironment proxies in these records to try and help solve this mystery. Okay, so what other trends did we see in molecular biomarkers at Trimposo, where we see the largest excursion that is the most distinct? What did we see during the Middle Holocene? So the first I'm going to show you are some abundances of macrofossils. So this is chironomid in head capsules for gram, and Daphnia, which are crustacean. We have their Epiphyia, which is their egg shell per gram. And we think these are a relatively rough indicator of productivity. And so what you can see is the abundances of these macrofossils increase in step with our decline in hydrogen isotope values. Then we looked at microbial biomarkers. So we looked at the ISO GDGT0 to quinarchaeal ratio, which are archaea produced structures. And we looked at the branch GGT3A and all of these molecular compounds, and particularly the ratio or the percentage as we plotted them, we believe relates to oxygen in these environments. And the thing that's really interesting here is that if we put these into a correlation matrix, they're almost perfectly correlated with our hydrogen isotope trends, which is absolutely wild when you think about how many independent proxies we have now in this lake system shifting in exactly the same way at exactly the same time. And by exactly the same way, I really mean in step with each other. Just to add one more thing in there for you to think about. We also looked at the amount of macrofossils from terrestrial plants that were in that system at that time, and that overlaid in terms of the peak appearance of those with our isotope excursion and our mid-chain waxes. And we think that's peak terrestrial greening. So when that landscape is really ramping up in productivity, both terrestrially and in the watershed. So all of these biomarker indicators point to a major change in the reducing conditions of the lake. And that means that the oxygen probably dropped in the lake and the reducing conditions changed. So you no longer, oxygen is your electron acceptor. And so you have to start looking at other ways to use organic carbon or other metabolisms that are going to start using that organic carbon because oxygen isn't available. And so we would argue, or I would argue, that these proxies show that the reducing conditions changed in this lake, partly due to an increase in productivity, so a loading of organic carbon that would use any available oxygen, but probably also due to an extension of the ice-free season and then a thermal gradient driving stratification of these lakes. And it's important to note that aquatic plants are going to grow in the Arctic summer, so the stratification would be occurring during the Arctic summer if we believe these trends are all related. So can major changes in reducing conditions of lakes influence aquatic plant wax hydrogen isotope values? Well, the answer to that question is really another question. what type of aquatic plant do you have? So in these lakes, I mentioned at Waxlips Lake that it was absolutely covered in a carpet of benthic mosses. And that was true at Trimfaso, and it was also true at N3. And so brown mosses are what the type of mosses are. And in this picture, I have just a picture of one Storphia exonulata. It is the type of moss at Waxlips Lakes. Drepanoclitus, Scorpidium scorpioides are the other types of mosses we see in these lakes. They're all brown mosses, and brown and sphagnum mosses have this documented relationship with symbiotic intracellular microbes, and particularly when you have a methane-dominated system, you can have methanotrophic bacteria harboring inside the cells of aquatic mosses. Methanotrophic bacteria take methane and then use oxygen provided by the moss and oxidize that methane. And methane hydrogen is typically about 250 per mil lighter than growth water. So that is already providing a potential source of extremely light hydrogen and a pathway for which it can enter an aquatic moss. Okay, so just to summarize what we see in the system is we see organic carbon loading, we see probably summer stratification. And if we believe that this is potentially a methane incorporation, then what we think is happening in these aquatic mosses is that actually that transition to depleted values in the middle Holocene is a shift towards representation of methane hydrogen away from meteoric water. If we believe that, then what these records are showing us is that across Greenland in this huge spatial range, in 3 lakes, which is a pretty big sample set for when you're talking about Holocene records on Greenland, we see these long periods of heightened methane production in Arctic lakes that today aren't methane producing and aren't stratified. So there's this clear increase in methane generation during the middle Holocene, and that helps sort of consolidate observations of methane concentrations and isotopes and ice cores during that time that isn't accounted for in the global methane budget. And we think that was driven by warming, but lag peak warmth because of how long it takes for the system to ramp up and catch up with the temperature. So for vegetation migration, for these soil development, for nutrient fluxes into lakes, and for those to become really productive. So all of this just speaks to the fact that there is potentially this huge flux of methane that is coming out in the middle Holocene from Arctic lakes, and that has really strong potential to portend a future for the Arctic, where many lakes that aren't considered in the methane budget because they're not stratified today, they're not methane emitting today, are going to transition to conditions that promote methanogenesis and methane storage and methane release. The one caveat to that, though, is that because we are seeing this in a moss biomarker, it's clear that some of it is uptaken into moss biomass. And so we don't know if these will act as a carbon source or a carbon sink or what the net flux will be, because it's a component of the carbon cycling in Arctic lakes that really hasn't been probed. Now, if I take that one step further, I had a plug really quick, which is that if you're interested in the details of the story, or you want to hear about the carbon isotopes, which we also have in a lot of this biomarker data, this paper, which is in revision at Nature Geoscience, is available as preprint. And I'd also love to talk to anybody about it further if you're intrigued or interested in that. Okay, so how should we modify our framework then in terms of thinking about plant waxes and their incorporation of hydrogen? Well, I think we need to add an entirely new branch off of our sort of framework for how we think they're integrating hydrogen, and put lipid precursors coming from microbial symbionts into that knowledge, and particularly for aquatic plants that have potential to be in environments with major changes in the reducing conditions. I'm going to really quickly jump now to where else can we look to develop new insights. So I feel like for the plant waxes, we kind of broke the proxy, but we're really trying to build it back up now as a methane biomarker, which is a totally new perspective in terms of biomarkers for Arctic lakes. Where else, though, can we start to develop insights about these environments? Well, I would say that plant biomarkers have a really long history, and they're one of the most common compounds and biomarker tools we use to understand these systems. But because we're doing that, we're missing all of these other perspectives when we keep using plants. And microbes, I'm really interested in right now because they play this really important role and hold a bunch of untapped information. in the isotopic composition of their lipids, which hasn't been probed to the degree that plant microbes have. They really modulate biogeochemical cycles, so we can really start to think and pick out the role they might have in these ecosystems. I've put on here compounds that are synthesized by a couple of very relevant soil bacteria and archaea that make structurally distinct lipids that are beautifully preserved on very long time scales. You can find these types of structures going back to the Cretaceous, and they can tell us a complementary story to what plant biomarkers have been telling us. They might tell us in their carbon isotopes a carbon history of the system, and in the hydrogen, they probably will tell us something about the water they were using. The reason these microbes and these tetraether lipids haven't really been incorporated into our biomarker tool set yet in an isotopic sense is that, for one, there aren't that many structures made by microbes that are extremely source specific, so they make a lot of compounds that a lot of other things make. And tetraethers can be the exception to that. But they're extremely challenging because they're such a big molecular compound. They're too big to fly in a traditional GC-IRMS setup. So instead, what we have to do is we have to break those ether bonds. So I kind of highlighted it here in my archaea isoprenoidal lipid, that carbon-oxygen carbon bond, which we can do, but it's really hard to do without also imparting hydrogen isotope effects. But once you've broken that bond, and if you can do it in a way that you can control the hydrogen exchange, you can liberate these fragments that are hydrocarbons that are stable, that have lots of carbon, that have lots of hydrogen, and start to look at the isotopic composition and how that really suppresses these in the environment. So the work I'm doing now is really trying to isolate these from these complex environmental structures, get them concentrated and pure enough for isotopes, cut them into small pieces to get the hydrogen isotopes and the carbon isotopes pretty reliably without prep chemistry fractionation. And here's just a really quick picture of what that looks like. It looks a little grainy on my screen, so I hope it's a little more clear for you. But that's me working on a MAT 253. I'm trying to get our sensitivity a little higher so I can use smaller sample sizes. I'm in my bios, I'm using hydrogen gas to break off an iodide that I used. So I use hydroiodic acid to cleave that bond. It adds an iodine to my compound, and I'm trying to bubble it off with a platinum catalyst and add a hydrogen from hydrogen gas on there. And what that looks like in mass spec is if you take these compounds, you break the ether bonds, you get 3 distinct biphytane peaks. And just a really, really quick spoiler alert, what we see in the archaeal lipids is a very, very strong correlation between hydrogen isotopes and growth water in cultures. And so that has a lot of potential to start thinking about, okay, well, archaeal lipids are found in both marine and terrestrial environments, and they go really far back in the geologic record. And so they have this huge potential power as the proxy. And so this is work that I'm collaborating on with Ann Pearson in her lab at Harvard and Will Levitt in his lab at Dartmouth. Okay, so from there, I was just going to really quickly tell you, in summary, where my research is pushing the field forward, and it's really in these prongs of geologic records and modern observations as tools to really understand how Arctic and global changes are likely to play out over the coming century. All of the things I've kind of talked about in my talk today and sort of the future where they're going really fit into this Venn diagram in those ways, but with really different goals. So one is developing hydroclimate and biogeochemical records through past periods of warmth. One is really probing this flux that we haven't considered in Arctic lakes and trying to get a constraint on how much carbon is exchanged in the system and how much could potentially be emitted to the atmosphere as these lakes start to stratify. And the final one is just this really extensive, novel, sort of broadly applicable utilization of biomarker isotopes from tetraether lipids, and really pushing forward both the analytical techniques and the applications on that front. And so with that as my final summary and running short on time here, I just want to really thank you for your attention. I want to acknowledge the Inuit people and their land on Greenland that they welcome us to every time we go to sample, and all of the support and collaboration that occurred along the way. And thank you very much.